So today we have Nadezhda Azbukina. Is that how you pronounce your last name?
Yeah, almost, yeah,
Almost
Azbukina.
close.
Yeah,
From Barbara Treutline's group in Zurich. And really, this is a continuation of something Dr. Fatima Sanchez Calleja began speaking to us a few years ago in this same series about different ways of implementing morphogen screening across many lines in human neural systems in vitro. And of course, this is something that we've had a lot of talks on in this group and are super interested in. So thanks for making the time and coming to speak to us.
yeah,
Take it
okay.
away. Thanks so much.
Yeah. So good afternoon, everyone. Thank you very much for the opportunity to present our work. I'm happy to do so, and I'm quite excited to share our recent work on human neural organoids from Troitline Lab. Actually, we're in Basel, so there's like one
Oh,
department of ETH in Basel. So
okay.
we sit in Basel. Yeah, just yeah, and okay. Uh, let's start. So today I want to show you kind of two examples of how in our lab we utilize high-throughput single-cell genomic screenings to learn more about morphogen effects on neural organoids patterning. And kind of let me start with the first project, which is dedicated to study systematic organoid patterning and its reproducibility. And this paper has been published online just before the Christmas, so it was a nice Christmas present that we managed to get it through. And also we contributed to the research briefing where we summarize our finding in a more concise way. So like a 2 pages short summary. So if you are interested in more details, so please check it all out. And let me start with a small introduction that human brain is an extremely complex organ which controls the way we think, move, feel, and react. And brain development and formation is a multi-step process, and it lasts till early adulthood. So it's kind of clear that the human brain function and development is very important to study. However, there are quite many limitations to the system, which includes inaccessibility of early stage samples, lack of experimental intervention, ethical concern. So that basically for sure brings lots of restrictions to study the system, and we mostly can do only observational studies. And here come our favorite organoids, which can fill this gap and let us a bit manipulate the early human brain development. And also if we form the embryonic body and wait long enough, we can see nice layers of neural progenitors and neurons forming this nice layer structure. And as we know, there are two main types of organoid protocols. First being unguided, when we just form embryonic body from stem cells and then bring it to neural induction medium and let it pattern by itself. So usually this type of organoids are composed of different brain region identities. And on the other hand, we have guided protocols where we basically, with morphogens or some other factors, try to guide the patterning of tissue into particular brain region. And usually these organoids would be enriched with particular cell types. And over the last decade, the neural organoid field has been kind of rapidly exploding and growing, and many labs are working on developing their own protocols targeting particular brain regions. And here you can see the table that Fatima nicely put together, summarizing main protocols targeting different brain regions like encephalon, midbrain, and so on. And you can also find this table as a supplementary table 9 in our paper. So yeah, if it's very useful for you. And what was particularly exciting to see when we tried to just have a quick glance on the table, that in order to target and get the same region, people actually used different neural induction approaches, different patterning molecules. So yeah, it was quite exciting to see that you can tackle the same problem from different angles. And kind of in order to systematically have a look at this, our lab previously, in collaboration with other labs, led an effort to integrate it all together in a human neural organoid atlas. So basically, they collected many publications and many organoid datasets until 2024 time point. And this is basically the result of their work. Here is a UMAP of more than 1 million cells from different studies of neural organoids. And here the cell type annotation is based on a marker gene expression. So it's basically agnostic to original human organoid protocol. However, if we have a closer look on which basically brain region people were trying to target in, we see quite interesting results. So for example, here, protocol 7, so basically 3 different publications use the same protocol trying to target the telencephalon. And you could see on the UMAP, these black dots basically would be these cells from this protocol, from this study. You can see that people use exactly the same protocol, but got quite different results. And the same picture would be with some other protocols. So basically, this once again highlights that there is a huge technical variability, that even if people use the same protocol in different labs, it can result in quite different patterning outcomes. And once again highlights the need of systematic morphogen studies where we control for lots of technical variables, because here we cannot really infer whether these effects we would have from different morphogens or just because the lab use different line or different neural induction-based medium. So and here comes our paper, which is mostly centered around 2 main experiments. So first of all was patterning condition screen, where we focus on screening many conditions, many morphogens, with different timing, concentrations, and combinations. And a second big experiment was more focused on reproducibility. So here we used a bit less morphogens, but more cell lines, 2 different neural induction approaches, and 2 different technical batches. And we've also done lots of fun analysis. So we did cell type composition analysis, we did morphogen interaction analysis, and regulon activity. And here I want once again to highlight, so this basically, the whole project was started and led by Fatima, and I joined mostly for the revision experiments. So yeah, this was a results of fun, long-lasting collab. Yeah, and I'm very grateful to work with you because this was really fun. And yeah, when we try to talk about morphogen screens, we are not the first one who thought about this problem and not the only ones. So over the last 2 years, there were several papers that have been published that kind of try to approach this problem. So first of all, this is Amin et al. Screen from Sergio Pascal lab, and Soraya Scuderi screen from orthogonal patterning screen from Flora Vaccarino lab. And they basically both sample later time points, so more than 2 months old organoids. Both use dual SMAT inhibition. And yeah, I will not go too much in details here because actually first authors already gave a talk at the seminar series. So please check it out. I think they will give you better review there. Yeah, and actually our study kind of complements this work because we account for some variables that the previous papers didn't account for, and we also used quite an early time point, day 21, because this allows us to both sample progenitors and neurons all together. And we also have established a systematic windows for testing morphogen effects. So basically each morphogen was probed at the same timing windows and in the 5 different concentrations, which were basically chosen based on the literature review. And yeah, after doing all of this systematic probing of morphogen effect, Fatima was able to collect like more than 100,000 cells. So it's a beautiful data set showing basically spanning covering quite a big range of central neural system progenitors and neurons, which also show quite a nice diversity. When we re-embed and recluster them, also we have peripheral neural system progenitors and neurons. So this is quite a rich data set to explore. And when we think about quantification, like looking at this from quantification angle, we would plot this cell type composition plots. So each column would be different condition as a control or different morphogen treatment, and they are basically grouped by timing experiments. So here we would have different, like morphogens for a different timing window, different concentrations, or combinations of them. And this is all like represented from the only one cell line has 3 cell lines, so for which we've done the most, basically like the most experiments with. And yeah, so kind of when we just look at this data, we already can see some interesting tendencies. So for example, and trends. So for example, this retinoic acid, if we increase the gradual increase concentration of retinoic acid, we would have a gradual increase of hind brain progenitors and spinal cord progenitors. While, for example, if we have a look at BMP for concentration steps, we would see that we have rather a binary kind of response. So this is a control, and this is a block of BMP4 with different concentrations. And we would see as soon as we add a bit of BMP4, we already have a shift of cell type identity, and this kind of doesn't matter that much with increase of the concentration. So this was quite interesting to see these trends. But I'm not gonna go too much into details. And if you're interested in this thread, so please check out previous talk from Fatima in July 2024. Yeah, and today I'm rather gonna focus on the analysis and data that we added during the revision. So since we sampled mostly the progenitors, we can infer the regulon activity. And a big shout out to Jasper. So he's a postdoc in our lab, and he basically developed this extension of scenic algorithm to connect the regulon activity to a morphogen. So basically, based on this analysis, we can infer this morpho-GRN. So gene regulatory networks, which are modulated by different morphogens. So how can we read this plot? So in the center, we have the morphogen. And each morphogen is linked to different regulons. And basically, each regulon is basically a transcription factor and set of genes which it regulates. And if we have a look which regulons are activated by different morphogens, we would see that basically we see lots of expected results. For example, lysinoic acid modulates activity of OHOX regulons. Sonic hedgehog kind of modulates activity of different ventral factors. So it basically reassures us that what we infer from our data has a biological meaning. And yeah, I forgot to mention that the color of these dots, of these regulons, would be the corresponding colors of the cell types in which these regulons had the highest activity. So, and in order to basically draw some kind of conclusion from this type of analysis, we've done the following thing. So basically here on this plot, we basically try to plot the correlation for each regulon activity with the timing on x-axis and to the concentration on y-axis. And so basically, yeah, here each dot would be the regulons which we see here, just plotted on x-axis the correlation to timing, on y-axis correlation to concentrations. How can we interpret it? It's basically if a regulon has a high correlation to concentration, this means that regulon activity would be increased when we increase the concentration of morphogen in the medium. And if regulon has a high correlation to timing, that means that basically regulon activity increasing if we add the morphogen a bit later. And if we just have an overview of this plot, we already see that different morphogens have different modes of action or like kind of modulate the activity of regulons quite differently. So for example, FGF8 regulons are responsive to timing. So you see kind of they're all around zero. However, if it comes to concentration, you see the nice kind of distribution. So basically, quite many regulons which are modulated by FGF8 would be responsive to concentration of FGF8. And for BMP4, we have completely opposite situation. So the correlation to concentration is kind of flat. So kind of it's a small one, so smaller in comparison to other morphogens, but correlation to timing is quite nice. So basically, as we also seen in the cell type distribution graphics, yeah, so concentration doesn't matter that much. What matters rather is the timing of addition of BMP4. So this basically gives us already lots of insights about modes of action of these different morphogens. Yeah, and also I forgot to mention, in case you have any questions, feel free to interrupt. Yeah, and then we can just already start the discussion. Yeah, and next step of the analysis, what we done was looking a bit closer to morphogen combinations. So here you can see this experimental grid that Fatima designed. So here we would see the probed anterior-posterior morphogens, dorsal-ventral modulators. And this is all results for Has3 cell line. In the gray, there is a single morphogens or control. So this in the middle, this is basically no morphogen. Let's say this is only XAF low concentration. This cell, for example, is XAF high concentration. So, and then the grid intersection would be like, for example, this is like low sonic, high XAF. So this is how you would read this plot. And when we have such a nice systematic experimental grid, what we try to do is basically try to measure interaction between two morphogens. So let's say we have a readout of how the first morphogen acts on the organoid and second one. And we have also the combination. So basically we check what would be the, if the effects of morphogen are additive, what would be the cell type composition, kind of ideal additive effects of morphogens. And we check how far away is it from what we actually measure. So if it's really far away, that means that morphogens acting are not additive. So basically there is some kind of so-called we can say epistatic interaction with them, synergy. Also, so basically the combination of them is not just a sum of what they act. And if we check the first example, like high XAF and high sonic, this basically shows us the highest like discrepancy between what we would expect and what we measure. And what we see that basically, yeah, let's say high XAF is like this cell type composition, high sonic is the cell type composition. And what we have when we add two of them is quite different. So basically we have unexpected cell type distributions, yeah, which basically tells us that, okay, yeah, this morphogen have some kind of interesting synergy effects between them.
And
And we've
Nadja,
done
which is that green fate? I'm trying to look at the map on the right.
this one.
No,
No,
the
this,
one that comes up. Yeah, exactly.
yeah,
That's this one here.
this is
Which
non-neural ectoderm. Yeah,
when you put them together. Okay, thank you.
yeah, exactly, yeah. And then, yeah, so this was kind of unexpected because like in the solo, they do like, yeah, telencephalon, some, yeah, neuroectoderm. Yeah, this will be more like kind of, I think, yeah, this will be a bit of non-neural crest and yeah, cortical hem, but yeah, altogether they do something else, which is kind of unexpected. Yeah, yeah. But some of them were kind of more linear. Let's say, yeah, this example, yeah, that basically XAF, low concentration, and cyclopamine, high concentration. Yeah, so this is combination, and yeah, and this is like solo XAF, and this is solo cyclopamine. So it's basically, yeah, kind of average of them. Yeah, yeah. So, yeah. And the same type of the analysis, the same type of experiment we've done also with WTC cell line. Also, yeah, measure this non-additive effect. And what was quite interesting to see that basically consistently high KIR for both of them cell lines kind of resulted in a mesenchymal cell fate, like basically the majority of organoids was mesenchymal cells. Yeah, which was maybe a bit unexpected, but yeah, also because we used minimal NIM, so basically like an Lancaster medium. Yeah, that's why we thought that, yeah, maybe if we use dual smart with a KIR, it should result in a more neural fate. And also if you just look with the bare eye, comparing left and right plots, you could see that actually they're quite different. So again, like cell line variability is quite big. And this all together kind of pushed us to think about the follow-up experiment, which is more focused on reproducibility in the morphogen patterning.
Actually,
So
before you
yeah,
go there, I would like to ask, maybe some of the non-expected results could be also because of the interaction of timing, because you are like studying the interaction of different morphogens, but also time, it matters. So whether you add it before or after the two of them also could explain that, no?
I think here basically Fatima maybe can better answer
Yeah,
the timing. Yeah.
I totally agree. And actually, we have another plot in panel T, I think it was, or the figure. I was actually just looking at it because it's a very good question.
What's the timing of this experiment? What time point is
so
this?
yeah, XAV 939 is added day 0 to 9. Like we always kept that constant for each of the morphogens. And then Sonic Hedgehog is day 3 to, I think it was 12, something like that. But basically this is kept constant. And yeah, like actually for XAV, like it's probably the one that's already priming for non-neural fates, like you're inhibiting wind. And then Sonic just comes and like completely messes everything up.
Yeah.
We actually looked at which of the two morphogens has the predominant effect, if it depends on which one is added first. And actually, it not always correlates with the one that's added first. And in this case, it was like something completely out of the blue. We found some other
This one, right?
examples. Yeah, so the one that's in bold is the one that's added first. So yeah, it's a bit like maybe hard to interpret everything, but the center would be the expected combination, and then each of the dots are like each of the morphogens alone. So XAV is farther away from the combination. That's like the predominant effect in that case, which makes sense. But then in some others, for example, I think down there,
Like here,
So it depends on the
right?
cell line. Which cell line is this,
I think it's
Nadia?
S3. I think
Because I
S3.
think one of the cell lines does respond more, like the first one. I mean, I don't think we can set a rule for this, but like we were curious also to see like if you add it first, then is the combination more similar to the one you added first? And it's not
Can I
always
ask,
like that.
do you have, just asking, probably you to know the answer, but do you have IPS data from those, like before putting them on the treatment? Because it would be very nice to see how the IPS gets affected by those morphogens, how the variability they have.
Yeah, we don't have that, but we were thinking about it during the revision. We just had to set on like a set of experiments that we could do. But yeah, I think it's a really good question, and it would be great to have that.
Great, thank you.
Okay, and we go back. So this was, here. And so as I mentioned before, that we like looking at all this data, we kind of started to think more about reproducibility, but more so-called like technical, but not maybe really technical variables that we need to kind of systematically probe. So one of them would be donor line. So also like different donor lines have different sex of the donor, also different methods how to get the cell lines. So embryonic stem cells versus iPSCs. Next would be the batch. So just a technical batch. If I do experiment today and kind of in a month, would I have the same results with this morphogen patterning? And also neural induction mediums. So we use mostly the first screen minimal NIM. But what would happen if we do the same morphogen perturbations, but with a dual smart base medium? So kind of we try to explore all of these questions. So we designed this experiment. For this, we basically narrowed down to 12 morphogen conditions. So first screen was like 97 conditions. Here we only picked 12, which kind of more representative. So there were only 4 morphogens in 2 concentration, lowest and highest from the first screen that was done. And also there was from the first screen here timing missing. So we also edited here. And also we took into account some technical variables. So we picked 4 different cell lines, male and female ones from embryonic stem cells and induced pluripotent stem cells. Also, we've done exactly the same set of experiment 2 times, like pouring a different cryo vial of stem cells, cooking medium differently, so like a proper different technical batch, and 2 neural induction methods, so minimal NIM, like Lancaster media, and dual smart inhibition media. So it's basically SB and noggin. And if you multiply 12 by 16, it's kind of 192 samples we have to probe, and we can't do it with a drop-seq based method. So that's why we went to combinatorial indexing single cell platforms such as Parse Bioscience, and basically they provide like a 96, like a kind of their kit allows to barcode 96 samples. We had 192, so basically each sample we had to combine 2 different cell lines in the one vial, kind of to be able to barcode it, and then we demultiplexed it later with based on SNPs from different cell lines. So after doing all this crazy stuff, we've got 192 samples profiled, more than 200,000 cells, and we've got this beautiful data set. And what was actually quite interesting to see here is that we've got this midbrain cell population, which we didn't get at all in the previous data, where we used only minimal NIM. So this was like including dual smart. So this was quite interesting to see that, midbrain cells actually need some kind of different base medium. It's quite hard to get them without any additions. And if we try to look at the reproducibility, so this was our main question. So basically we quantify the cell type composition. So in the columns, you would have different morphogens. And then batches are in rows. So row 1 and row 2. And left-hand side is minimal NIM and right-hand side dual smart. So basically, if you want to check whether our batches give reproducible results, we need to compare 2 rows. And kind of if you just look at this, looks kind of quite similar. So this was for each one cell line. And if you check other cell line, like WTC, kind of also if you just skim it through, like looks kind of similar. And the same with WIPJ2 cell lines, and H9 actually is the same. We just didn't fit nicely into the plot. So however, we also wanted to quantify it. So basically try to get for each of the morphogen treatment and a cell line kind of a metric to say, okay, how reproducible this condition was in our hand in our experiment. So for this, we come up with two different metrics. So first was this KLD, so Kullback-Leibler divergence. So basically, to put it simple, let's say we have a cell type distribution for one batch and for another batch, and we basically compare the cell type distributions. And the smaller the metric is, the more similar conditions are. So basically, we kind of really want that it's all around 0, and that's what we've got. So basically here, for each of the morphogen condition, I scored this divergence between two batches and plotted it by cell lines. And you can see, first of all, that here that most of them are kind of low, so almost at 0. True, for each of the cell line, we see some conditions which were a bit less reproducible, but overall the trend is clear. And also what was quite interesting to see that the degree of variability of patterning is kind of the same. So we cannot say that, okay, like iPSC cell lines are more robust in patterning or robustly respond to morphogens than ESC cell lines. Or like female cell lines more reproducible than male. Kind of, at least in our sample size, I know it's kind of small, but at least we couldn't see these trends quite clear. However, this kind of metric has a bit of a drawback because it relies on cell type annotation. So let's say tomorrow I want to change cell annotation, say, okay, yeah, I want to annotate it more deeply, certain progenitor cell population. And this whole metric will change. That's why we went to a more distributional metric called E-distance, energy distance, which was introduced in this paper. So basically it takes into account so let's say when we embed the cells in a low-dimensional space, such a PCA, and then we have a distances between organoid 1 and 2, and also like basically it accounts how far away these distributions are from each other, and also take into account how spread they are. And again, the smaller this metric to 0, the more similar our conditions are. And we've got more or less the same profile of the results with this distributional metrics, basically kind of reassuring us that conclusions we drew with this metric was, hold it and are correct. So
Can I ask a question?
Yes,
so,
true.
When you're looking at these two metrics, KLD and E distance, let's say in H1, there's one that's the highest. Are they the same condition? Do you get similar results in the E distance analysis versus the KLD analysis?
I think they're overall correlated, but I don't have a plot.
Okay.
But usually the outliers kind of would be the same. So usually it's KIR minimal NIM. So basically when we go into this off-target cell fate, like mesenchymal, there is basically quite different from organoid to organoid. So usually this is basically all skewed by these conditions when we have off-target fates.
So are they the same conditions in all cell lines that are outliers?
This
cool.
I don't remember, to be honest. I do not know. I cannot answer you, but it's a good question, actually. Yes.
Yeah. So what are these, and what are these, basically?
Yes,
right?
exactly this. I mean, I think we have it's in supplementary table, but yeah, I would need to check. Yeah.
Okay, thank you.
Yeah. Yeah, and once we kind of looked at the batch reproducibility, we also have a look at the neural induction medium. So comparison whether we have the same results of a morphogen treatment if we use minimal NIM or dual SMART. And basically this base medium was used for first 2 weeks of organoid patterning. So, and when I just plot this cells colored by which base mediums they were in, you already can see quite clearly that these cell populations do not kind of overlap or have a very minimal overlap, which already tells us that yes, there will be for sure differences in how the morphogen affect cell fate depending on the base mediums that we use. And when we also try to quantify what would be the proportion of progenitor cells, neurons, and off-target populations in this organoids, we actually, as expected, nicely saw that dual SMART increased the proportion of neurons, mostly because all of these off-target cells, which we have in a high KIR concentration, actually got a neural fate. and we have much less off-target populations. And yeah, actually, there's also, I forgot to add, it's statistically significant. So one could think, okay, dual smart is the way to go. But yeah, however, when we check on the reproducibility, especially across different cell lines, we would actually see that minimal NIM would have quite like more stable, like more similar patterning outcome than with dual smart. So here, for sure, we would say that it has more outliers, but they're mostly driven by the KIR, like a high KIR concentration. But on average, like kind of not average, but median is much lower with both metrics for minimal NIM than for dual smart. And yeah, usually like it's mostly driven by cell line differences there. So that like different cell lines actually respond more similar in a minimal NIM than in dual smart. And if we actually plotted split by morphogen treatment, so here you would see that the colors of dots would be morphogen treatment, and the color of a background box plot would be the base medium. And here, this is most reproducible kind of conditions. Yeah, you would see that most of them would be green. So these are all from minimal NIM, and only like 1 to 3 like from dual smart. And also if you check especially the control cells, kind of we would have more kind of from different cell lines, controls would look more similar in minimal NIM than in dual smart. So basically this was mostly driven that some cell lines, yeah, started to acquire kind of more posterior fate in a dual smart, yeah. So, and then when we try to kind of look at this all from the kind of general overview perspective, so kind of to have some kind of a roadmap or compass of this morphogen effect, what we try to do is basically to map our data to dorsal, ventral, anterior, posterior axis using different gene sets. So basically scoring how far away this condition with its more dorsal, more ventral, anterior or posterior. And we get this type of plot. So if we have a look at it, basically here on y-axis is a dorsal ventral. So here is more dorsal, here it's more ventral. On x-axis is anterior, posterior. And this is minimal NIM conditions, and this is dual smart. And each dot would be one condition and colored by morphogen treatment. And shape of this dot would be cell line. And what we see nicely here is certain kind of trends that for some morphogens, such as like high sonic or retinoic acid, you would see that different shapes come together and rather group by color. So basically, it doesn't matter which line we start with. If we add retinoic acid in a low concentration or in a high, it would basically shift to posterior fate. And if we add high concentration of sonic, it will be really all ventralizing. But if we take a bit like some other morphogens, for example, low sonic or low FGF8 or even high FGF8, you would see that rather kind of conditions are grouped based on shape. So like squares are together or circles are together. So basically this patterning condition wasn't, let's say, strong enough. So, and here cell line internal bias wins. So kind of rather it stays more different from each other. And the same kind of we observe with dual smart, just we see that actually KIR gets neural fate, so basically it's all kind of nicely together, and also early kill timing, but for example, late KIR timing, this dark red or yeah, dark rose, it's all scattered across here. So basically, yeah, it's matters quite a lot how we add the morphogen at which concentration. So, and I think, yeah, it can really help to design a kind of predict, like, let's say, okay, if we use this morphogen and if let's say other 4 cell lines reacted the same, probably other cell lines, we can't predict for sure, but probably will also react the same. Yeah, well, for example, other morphogen treatment, like different time point or different concentration, yeah, doesn't override the internal cell bias. Yeah.
Can I ask you a question?
Yeah,
just
sure.
to make something clear for me.
Yeah.
For example, if you look at this minimal NIM condition,
yeah,
and you look at this sort of like middle posterior position,
yeah,
the lines and the conditions are spread across a dorsoventral gradient. Are these spinal cord fates that we're looking at when you say dorsoventral?
So I think actually we don't have many dorsal ventral spinal cord markers in this gene offset that we're scoring. So it will be mostly like telencephalon. I think I have a set of gene actually in the supplementary slides.
Not to worry, I was just asking, yeah.
Yes, this one. So basically, this was a gene that we tried to score for dorsal ventral. So it's not specifically for spinal cord. Yeah, so yeah, I think it's more for the major part of the brain.
Cheers. Thank you.
And yeah, and this all brings me to the conclusion of the first part of the talk. That yeah, basically our main conclusion was that like neural induction protocol and choose of cell line will significantly influence the patterning outcomes. Sex of cell line donors and the origin of cell lines did not impact, at least in our head, the technical batch reproducibility of patterning. And kind of we saw the tendency that higher morphogen concentration will more likely override cell line specific biases and will result in a more reproducible patterning across different cell lines. Yeah, and this brings me to the second part of the talk, where we more discuss how can we try to design new cell types, having all this information and all this knowledge that we get from previous papers in mind. And this basically what I'm going to present is a part of other paper where yeah, I was very happy to kind of collaborate on it with Jisong and Sichuan, who were first co-authors on this paper. And yeah, this paper more focused on a posterior brain atlas. And one part of this that we try to generate, like do morphogen screen to generate more posterior fate cell types. Yeah, and then kind of to give a bit of a introduction to this part of the story, yeah, we all have this, again, this human neural organoid cell atlas, which was done yeah, by also Jason in our lab. And basically, in order to try to get a feeling where are we as an organoid field, which cell types can we get, which not, they mapped this integrated organoid atlas into the primary human brain data, basically Brown et al. From Linarson lab, which spans kind of a bit further away than first trimester of a developing human brain. So they mapped this data to this data set. And here you can see the results of this analysis. So basically, this is a UMAP of a primary brain data. And it's colored by presence score. So basically, whether the certain cells of a primary brain, we can find them in organoid data. And in blue, this is parts that are enriched in organoid data, so quite dominant. And in red, there's part of the primary brain which are not either not present or not a dominant cell type in the organoid data sets that we have in this integrated organoid data. Actually, I would get rather laser pointer. Yeah, so this red part we are missing, so we are not yet able to make them well in a dish. And when we try to check what are these parts of the brain, so basically plotted split by brain regions, dissected brain regions of this atlas, it's mostly actually so-called posterior part of the brain, like thalamus, midbrain, cerebellum, so other part of hindbrain. And so basically we try to get this as an opportunity to try to design kind of a morphogen screen which would expand target with the diversity of this posterior brain part. So for this, we basically took a protocol like from Chen et al. From 2016. This was a baseline protocol. And we know that it generates midbrain, like ventral midbrain. So basically, as a protocol, first week, it's more like a neural induction with sonic and FGF8. Then second week is a KIR, more like a patterning. And from second week, later on, it's neural differentiation. So there's more like neural growth factors, BDNF, GDNF, and others to kind of promote the neural maturation. And here we sampled a bit later time point, 5 weeks, to kind of have a bit more of a neural diversity there. And the major patterning, what we've done, and major patterning perturbation and morphogen grid we've done on the second week of organoid development. So basically to target this patterning window. And yeah, we've designed this 48 conditions using 10 different morphogens and many combinations of them. And yeah, here big shout out to people who helped me to execute this experiment. Bijan helped with culture, and Gosha, Rivoko, and Sichuan to help on the day of experiment with sample fixation, because it was like a factory to basically preserve 96 samples in one day. And then for the morphogens that we used some dorsoventral morphogens, which is more BMP4 and sonic hedgehog, and anterior posterior one, retinoic acid, KIR, FGF8, FGF17, FGF2, and arspodin 2 and 3. So And after sequencing all this data, we basically got like 170,000 cells from 96 organoids. And what we see here is basically the UMAP of the data. And in black, the colored control cells, which are present in control organoids. And in gray, cells which would be present in whatever treatment we have. So basically, just looking at this plot, we see that we expanded the diversity of cell types that we see in this organoid protocol quite a lot. So quite many cell types are not present in the control. And then when I try to annotate it, it also shows remarkable diversity. So we have a ventral midbrain part, dorsal midbrain part. Red shades would be like cerebellum pons. Brown is a dorsal medulla, and yellow is a ventral medulla part. And when we try to check like at what will be the cell type composition across different protocols, so we would see that here in a column, we would have separate conditions. And this is a cell type composition for each condition. And this would be which morphogen treatment they got. So that basically it's kind of color-coded. And you can already, if you just look at this plot, you can see that some conditions were more like diverse. So you have more cell types, and some conditions are dominated by one or two cell types. And basically, kind of, we wanted to some kind of quantify it, say, okay, this condition is more diverse than this one. And what came into our mind is basically the metrics that ecologists use to quantify ecosystems. So like what is the species diversity in this ecosystem? And we use it what is like in the context of what is the cell type diversity in our organoid. And this would be the Shannon diversity index. And the higher it is, the more diverse it is. So let's say if we check on this higher, the most diverse condition is condition number 6. So we check it, and it's really like basically slices of cakes of different cell types. And if we check at the condition number 4, less diverse one, this will be this one. So basically dominated by 2-3 major cell types. So yeah, basically this metric kind of shows us, okay, if you're interested in particular cell type, what would be the most condition which is like robustly and mostly generated with a more cleaner cell type composition. And also kind of to the same flavor of a previous paper, what we've done is try to map it to anterior posterior dorsal ventral axis. And here would be the mapping of a control condition. So let's say this is dorsal ventral, anterior posterior. This is where we are, kind of ventral, as we expect. However, if we add retinoic acid, so this is like the plot is like a difference in density. So green will be this, in this condition, it's enriched, this part of this anterior posterior dorsal ventral axis in comparison to control, and dark gray would be decreased. So basically we see that retinoic acid kind of shifted towards posterior, as we expect. BMP4 would be more dorsal. And if we add it together, we actually go to a dorsal posterior fate. So this is like quite a nice example of quite additive effects, let's say, of morphogen, which is like the way they would act, the way we expect. And however, kind of it's still not answering all this analysis, the questions that we basically put in front of us, that whether we can generate new cell types that were not previously generated with organoid protocols. So for this, we basically mapped our data into also Brown et al. Atlas. And this the results that we've got. So basically the blue, kind of what we are enriched in, is more or less here in this part of the UMAP. And if we color it by dissected region, we see that it's mostly represented by midbrain, hindbrain, like cerebellum, pons, medulla. So basically we are in this UMAP where we wanted to be. This was quite nice. And also if we basically compare to the organoid atlas, so basically from presence score of a morphogen condition, so from our data set, deduct or subtract presence scores of the organoid atlas, we would have this delta kind of. And basically what we gain in comparison to all other organoid protocols that were present in this atlas. So you will see this dark parts that basically mostly where we gain. And if you split it by brain regions, we would have this different cell types. So like the particular clusters of primary atlas, this way basically we were able to get. So it's like from cerebellum, LHX9 positive neuroblasts and neurons from pons, glutamatergic neurons from medulla, glutaminergic neurons from medulla. Yeah, so this was quite interesting to see. And basically with this, it kind of allows us to come up with certain protocols or suggestions that, okay, this condition will result in this cell types from these brain regions. And this again brings me to the conclusion of the smaller part that, yeah, this combinatorial morphogen screen actually allowed us to expand posterior organoid protocols. We were actually also able to generate some novel cell types in vitro, which were not present before. Yeah, and to kind of finish up this talk, I wanted to open up with like a small outlook that, let's say, we've done several morphogen screens, other labs also have done them. So as a field, we already accumulated more than a million cells, I guess, per job with different morphogens. Can we some kind of leverage all this data together and learn something from this? So in this direction, basically, like Dominic and Jonas went into. So they basically trained the deep learning model called self-flow on this organoid data. So they collected this from three data sets, all these different morphogen perturbations conditions. And they trained the deep learning on it. And so the model was able, so when they made sure that the model is able to correctly predict what is already known, they basically generated all the unknown combination of morphogens that they could come up with. So basically trying to predict, okay, this morphogen combination we didn't see before, based on what we know already from this data, what can it look like? So for this, basically they have this now virtual organoid dataset. So with like 23,000 protocols with the different baseline mediums. And yeah, it's more like a 70 million cells. And then, yeah, if you look deeper into there, they suggest that, okay, if let's say, yeah, we have a combination of this morphogen with this basal media, with this culturing condition, we should get more like a dorsal telencephalon cell types we didn't see before. Or let's say, yeah, if we have this combination, probably we would have more like a pons or medulla sulfates. So I think it's kind of a nice approach to think about, like a lab in a loop, so that kind of this model can help us to guide and design new experiments. So let's say, yeah, if you want a particular sulfate or particular cell types, what can be our starting point to start with, and like kind of think in this direction. And yeah, with this, I want to thank you very much for listening and for coming to the seminar. And then for sure, thanking Barbara and Gray, like my supervisors, for great supervision. Fatima, Jason, and Sichuan. It was really fun to work with, and it's all like a collaborative efforts. And yeah, I have an honor to present it now today in front of you, and also many other people who were involved, yeah, in planning, execution of this big experiments. Yeah, and I'm happy to take questions.
Thanks. A lot to think about. Please jump in, everybody.
I have a million questions, but I think I will try to be to to keep just brief it
Yeah, sure.
Okay, just thinking about this last one,
Yeah.
this outlook. How does this model accounts for donor variability if it does? Because it's very interesting to actually try to benchmark in the lab whether this is happening or not.
Yeah, actually, this is a bit of, I think, of a drawback here, because I don't think they accounted for cell line variability, because also for this data set, they only took the first part, not this reproducibility of donors. And each of this basically used a bit of different cell lines. And I don't think it was like a variable in the model. I can't say 100% for sure, but yeah, I don't think they really explicitly accounted for this. So yes, this will be, I think, quite interesting to include different donor lines as well, yeah.
I can continue if no other.
Yeah, sure, go ahead.
Go for
Yeah,
it,
so
Pavel.
I'm
Go.
happy. Yeah.
Going back to the minimal name and the dual smart
Yeah.
comparison. By the way, I
Yeah.
think the overall work is amazing. I'm just
Thank you.
it's amazing.
Yeah, I'm happy if you
I
like
was
it.
I was wondering if
Yeah.
you have actually checked
Yeah.
what are the regulons that are affected by the two different medias, because I'm sure that the efficiency you get for producing neurons or NPCs can be triggered by already what's in the control situation or what regulons are more kind of affected by the media, no?
Yeah, exactly. So this is basically more or less figure 5 of our paper about this. So I can try to show it now. Yeah. this one. So basically, this is minimal NIM and this is dual SMART. So and then you can basically see here, how much is regular response to concentration as well. So some regulon are the same, some are not. And here basically, the plots that, what would be the activity of this regulon in the two different basal mediums across different cell lines here. So we have some consistent regulons, but also some of them are variable. For example, here this this is control, this is low Kir, and this is high Kir, and this kind of sand color is basically non-available because we don't have neural fades there. So, and you basically see that, it's activated, but not a lot. But in dual smart, you would have it like more that it's activated in a control, but not in a Kir because it's posteriorizes. So this example, or for example, also Sonic Hedgehog, in minimal NIM, it's kind of this regular activity stays constant, but in dual smart with a high Sonic, it increases the activity like RFX2. So we have different examples of these regulons, that can explain. And bunch of regulons actually, for example, this gray one, so they were not present at all in minimal NIM, but were present in dual smart. And they're mostly like this, I guess neural fate regulons, because we don't, in the minimal NIM, it's basically all goes off target,
That's very cool.
Yeah.
Maybe I can ask a very general kind of question.
Sure.
You've raised all kinds of points. So I think that's why we're all hesitating a bit to know how to really comprehend it. So maybe one way of thinking would be you've sort of mapped the fates onto a kind of, I don't know whether you want to call it an end stage, but to a fairly differentiated state, as I understand it. But I wonder whether to solve this kind of problem, that what we really need to do is to actually map the fates out onto lineages so that we see a series of decisions. And maybe I'll just leave it at that. In the cell flow example, I was sort of trying to grasp whether that was what that approach was doing. My synapses had been depleted by that time.
Actually, I do not think that they would actually capture the lineage itself, but I think I know what you mean. I think maybe one can some kind of track it down if we say, okay, we have this timing. It was here, so I need to go to presenter mode. So for example, here we have this timing experiment, right? So this is a control. This is when if we add retinoic acid very early, like I think Fatima can help me more what was the exact timing window there, but like this window, this window, this window, this window. And basically you could see that, if we add it very early on, it all goes to basically a non-neural fate. But if we add it a bit later, we would have certain fate emergent. And if we add it really late, that, I guess it's also a bit of disbalance of a fate there. So kind of this, I think it's also what kind of tell us that different morphogen would have different sensitivity window, which we also, I guess, expect from the development. What we know in development is they all kind of, it's basically an orchestra of this cascade that first goes this one, then this one, and lots of these feedback loops. I think what might be actually quite cool that maybe, because here it's like a different experiments and a different time points. So might be cool is basically, you add morphogen and you sample basically the same batch day by day.
Right.
And maybe even the multiome. Basically, you see that, okay, how transcriptome changes, how epigenome also changes. This, like how the chromatin accessibility changes. So I think this might be actually quite cool, but also very expensive to do. But maybe one can do it for really like small windows and nicely dense sample. And really trying to dig deeper what exactly happens when we add this morphogen.
We did want to do that actually. Sample right before the morphogen and then right after the morphogen. For example, like related to Paul's question before, one thing could be doing like sampling all the cell lines at the iPSC stage. But then we were also thinking about like where are the cells, like what are they becoming at the time when you add the morphogen? Like what is expressed? Like we would have transcriptome data, so we cannot tell about like, I don't know, active receptors maybe that are different between the different cell lines. But I mean, there are lots of things that you can do, and definitely, yeah, the lineage thing. I think it's a really good point, and maybe we can. I mean, it's not my area of expertise at all, but like with all these computational methods that people like Jonas are developing, we can maybe put together all these data sets. The only gap that I feel like we have is that we are all doing things in different like conditions. So even if we use the same morphogens, there's always going to be a lot of noise. And that's why we ended up doing this and throwing so many morphogens, trying to keep like stable conditions and just to see like to have like continuous measurements and be able to like predict later. Because otherwise it's impossible. There are too many variables.
Yeah, I agree.
I was thinking two things out here. It would be amazing to have one cell line, transcriptome one cell line, and predict what is the best treatment for this cell line to get whatever you want. That would be amazing. And the second thing I was regarding the anterior, posterior, and the dorsal ventral plots. Obviously, if I have understood right, you use defined markers to define those, right? Or am I wrong?
Yeah.
Okay.
So for dorsal ventral, yes, and for anterior, posterior, this was a bit different. So basically, for anterior, posterior, this Jusong developed this linear model when you basically 1 would correspond to telencephalon, like 2 to diencephalon, 3 score 3 to midbrain, and 3 more than 3 is more like a hindbrain part. So, but basically, he was trying to like get the Brown-et-al data on the radial glia, and basically trying to predict from transcriptome what is the cell type label, and basically also kind of have this hard score on this part. But dorsal ventral, we just took the gene scores, yes.
What I mean, and that's a question that I leave it up here, is do the markers that for a certain time point define those axes are valid for later time points? That's what...
Yeah. It's an absolutely fair question. And I think it's actually also quite a lot there is still unknown. Like, to be fair, even if we check the Brown-et-al data, like you do not know where it's actually sampled on this dorsal ventral anterior posterior. These samples are usually very, very precious. And this also, I think there are a big question here, how to harmonize all these time points. Let's say, okay, we have this human brain development. And then we also have mouse embryos that basically, I guess people already sequenced all through in all different axes, but how to connect it to our real time point of a human development? And would be markers be the same? And then here comes organoid, like 10 days of organoid. Where is it in this trimester, like in the real week of development? So I think it's very, very tough. Yes, I agree. And these markers, I think they kind of tell us certain directions, but it could be, if add some other markers, it would look a bit different. Yes.
Anything else, guys? Well, thanks so much, Nadja. You've given us a ton to think about, and very, very interesting to keep seeing the sort of scientific and investigative flow across your own work and so many that we've heard from here. So, and I'd also like to sort of second what Fatima brought up earlier, is that if we can start to get a few more of these data sets in, maybe in one of the future sessions, we could do kind of a data-driven round discussion about all these things, where we could each of us bring in maybe some examples as well as questions that we could look at in the data sometime soon.
I want to second that, Carlo. I think the reason you're hearing this sort of silence here is because we're all sort of thinking. And that in the traditional way of talking about science, we have these one-off seminars. But actually, Carlo will post the talk. So we have an opportunity to listen to you again, which in my case is absolutely the same. Well, no, not really. It's just that there's so much data. So it's not so much that you're so fast as that we're too slow. And then I think, if I can just sort of speak to the point that you were making, Carlo, if in some format we can return to these issues and discuss them again, that would be, I think, really helpful to the whole field. Thanks so much for talking.
I think there's definitely a critical mass of data out there now that if we got it together, we could really dig in deep. And if we do just a little bit of work ahead of time to formulate some questions, grab some gene lists of interest, and it would be a blast. So we'll work toward that.
And let us know if you need any help with data upload or something. Or
Yeah, to tell you the truth, I'll ping you guys because, for this one data set or two data sets wouldn't be a problem. But as we go forward, the more we can incentivize researchers getting their own data there, the more realistic it will be to continue this, because the data production is of course only increasing, so it's hard to keep up. So
exactly.
I'll ping you guys and we can plug in, get some of this data in together.
Perfect.
Great. Thank you guys so much.
Thank you.
And we'll see you soon.
Thank you, Carlo.
Thank you.
Thanks so much. Bye bye.
Bye, guys.
Bye.